introducing an antibody drug candidate. My name is Maxine Chen, and I am the Marketing Liaison for Discovery Biology Services at GenScript. Today, I will start today's webinar with a brief introduction on GenScript and the services we offer. Next, I will introduce our Discovery Biology Services, which are relatively new at GenScript. Then, I will introduce Dr. Frank Fan, who will present on the process of producing an antibody drug candidate through antibody engineering and review some of the related services that GenScript offers. If you have questions during this presentation, you can email Dr. Fan directly at frank.fan@genscript.com. In addition, this archived webinar and links or downloads of any of the associated materials presented today will be posted. A little information about GenScript. GenScript was founded in 2002 as a provider of gene synthesis. Since then, we have grown to offer our customers quality bioreagents and catalog products. And since 2009, GenScript has become a one-stop service providing customers with full drug discovery capability as well. GenScript is cited in over 9,000 peer-reviewed publications where our services and products have helped move research forward. Our Discovery Biology Services offer our customers a seamless experience where we can provide all the services required for the entire course of lead identification and optimization. A customer can send us the sequence of their target, and from there we can generate an antibody candidate, test it for functional activity in vitro, and perform in vivo bioanalysis as well. To highlight some of our key offerings, our antibody and protein engineering services include generating single-domain antibodies, where we are the only CRO to offer this as a service. We are also capable of downstream engineering, such as antibody sequencing, affinity maturation and humanization. Our humanization service guarantees the affinity to be equal or better than the parental antibody. Our in vitro pharmacology services offer full cell line engineering capability, where we can generate custom stable cell lines with our lentiviral-based cell power platform. We have an outstanding track record of generating stable cell lines for difficult-to-express transmembrane proteins such as ion channels and GPCRs. We also provide CRISPR-Cas9-based knockout or knock-in cell line development and provide cell-based assays and ion channel and GPCR assays for functional testing of these custom cell lines. We also offer popular mouse tumor models, including subcutaneous xenograft, an inexpensive and quick way to test compounds. We offer orthotopic models, allowing you to study tumors in their specific organ microenvironment. For orthotopic models, we also offer the ability to perform bioluminescent imaging with our luciferase tagged tumor cell lines. We also offer syngeneic models for in vivo study of potential antibody drugs against tumor growth. And now I will introduce Dr. Frank Fan, who is the Director of the Antibody and Protein Engineering Department here at GenScript. Dr. Fan is a human B-cell immunologist specialized in transplant immunology. Prior to joining GenScript, Dr. Fan was a senior research associate at the Hospital for Sick Children at the University of Toronto in Canada. His research was the first to demonstrate that antigen-specific antibody tolerance can be intentionally induced in humans, and this work was published in Nature Medicine. He has also developed new techniques for studying applied immunology, which include a novel live cell fluorospot assay and a highly sensitive single-cell RT-PCR system to enable the amplification and sequencing of the variable heavy and variable light regions of the receptor of a single B cell. Bringing his expertise to GenScript, he has strengthened our capacity to offer highly customized antibody libraries, including a synthetic phage library based on alternative scaffold proteins and monobody and minimalist phage libraries based on binary coding. Dr. Fan holds both a PhD from the Graduate School of Biomedical Sciences from Hiroshima University in Japan and a medical degree from the Faculty of Clinical Medicine at Xi'an Jiaotong University in China. And now I will turn over the presenter controls to Frank.
Good morning, everyone. Thanks for coming. I would like to take this opportunity to introduce the general process of early antibody drug development and also share with you some of the tips and experiences in this industry. For the overview of my talk, I will first briefly introduce the challenges in the field and then talk about the standard discovery process with emphasis of antibody molecule engineering and its QC assays. In the end, I will briefly introduce the capability of our antibody engineering services and then take questions. As you all know, biologics drugs is very hot in the past few years, led by antibody drug and vaccines. RNA interferencing or cell-based assays are still mostly academic, and the gene therapy field has encountered major biosafety concerns. In the past decade, antibody therapeutics has been proven to be clinically effective for disease areas such as oncology and immunology. The antibody approach offers a greater target specificity as well as a new way forward with previously challenging targets, such as ion channels and GPCR. Currently, antibody drugs occupy 1/3 of newly approved drugs every year and also occupy about 50% of drug candidates in the pipeline of most biopharmas. There are at least a few hundred monoclonal antibodies being developed globally. The most obvious challenges in antibody discovery is the human immune response to an animal-derived antibody. Even when the antibody is successfully humanized by proper antibody engineering, it may still lead to anti-idiotypic antibody at clinical trial stage, which may impair the efficacy of the drug. Although much better than a small molecule, antibody drug still has off-target issues sometimes, and even renders undesirable side effects in the worst case. The high production cost of an antibody urges the need to wisely select the lead, which has good odds to be highly expressed, stable, soluble, and does not aggregate. To address these challenges, discovery teams need to have sufficient experience and expertise and do enough homework in the right way. Years before you can even dream of a clinical trial, you need to seriously think over every kind of decisions you will make, such as whether the target is sufficiently validated, which format of antibody you are going to choose. Is it a conventional IgG, Fab, rabbit, or camelid single domain antibody? What lead criteria need to be met in order to move on to the next milestone? And the million-dollar question, will it work in humans? Today, we will look at a key preclinical stage of the drug discovery process, from initial target identification and validation through assay development, hit identification, lead optimization, and finally the selection of a candidate molecule for clinical development. So the very first step is to select a target. There are more than 2,000 drug targets available, and there are dozens of top targets which competitors may already have a later stage candidate. A smart decision of a promising druggable target can make all the difference between success and failure. A few very important factors to consider include whether manipulating the target actually affects efficacy, safety of manipulating the target, and whether the target disease make commercial sense to develop an antibody drug against. A solid step to finding a good drug target is to do an initial data mining of literature, patent information, and publicly available gene, protein, and phenotyping databases. Alternative approaches such as phenotypic screening using high diversity antibody library may also prove to be valuable. Human genetic data which correlate to diseases is also a gold mine to explore. In 2014, a number of promising targets like ZNP8, now 1.7, and APOX3 emerged from loss of function mutation in patients. Of course, it is important to consider if the target by its nature can be targeted by an antibody. Developing an antibody toward GPCR and ion channels has been always considered a difficult task due to poor accessibilities on the cell surface. Immunology of targets like cytokine, cell surface receptors generally have good accessibility when antibody and a high binding affinity antibody can be readily generated. One interesting topic in recent years is the ability of cavalate single domain antibodies to cross the blood-brain barrier, which gives the possibility to treat diseases in central nervous systems using small domain antibody modality. Single domain antibodies are easier to engineer to adopt unique features through constructing a multi-specific antibody. Developing a new drug from the original idea to launch a finished product is a complex process which can take 12 to 15 years and a cost in excess of $1 billion. The idea of a target can come from a variety of sources, including academic and clinical research, and from the commercial sector. It may take many years to build up a body of supporting evidence before selecting a target of a costly drug discovery program. So the second important step is to fully validate the target candidate in-house. With the greatest expense of developing a new drug being associated with clinical trial, pressure on the discovery process to identify the lead candidate with a higher likelihood of success has increased considerably. This requires ability to make increasingly informed decisions about the individual leads at earlier stages. Multiple comprehensive validations with careful data interpretation are needed to reduce the risk of a possible expensive mistake in the downstream process. Using clear-cut approach such as efficient gene knockout or knocking technology to validate target process in vitro and in vivo will be a smart idea. Brand-new technologies like CRISPR-Cas9 may be useful in studies to win support for the R&D team. Once a target validation has been solidly conducted, there are still many questions that need to be carefully investigated. The very first thing to consider is which is the best antibody platform to move forward? Which species of animal to use? Conventional or heavy chain antibody? Rabbit or human phage display library? Or even a conventional approach like synthetic monoclonal antibody library? appropriate in vitro screening assays established already. Have the appropriate in vivo pharmacokinetic PK or PD models being established. Tissue distribution studies to evaluate PK may require evaluation in several species. Efficacy endpoints to evaluate PD also need to be pre-planned. Even before animal immunization, plans and a preparation of multiple screening methods need to be in place. Reagents for assay development and screening needed to be purchased or developed. Proof of principle efficacy assays such as ELISA and functional assays such as ADCC or CBC or proliferation assays may need to be developed and validated. Animal efficacy models will provide most robust data than in vitro assays, but collecting and analyzing data requires expertise to ensure the reproducibility of data between studies. In the event that a target protein has poor cross-species conservation, strategies for evaluating non-cross-reactive antibody may need to be considered. For example, generating a surrogate antibody or establishing a transgenic mouse model to express a human target. For lead identification of therapeutic antibodies, there are a few must-dos to validate a lead. The first is specificity, which can be established by determining their cross-reactivity with human tissue organ sections unrelated to the targets by using radiolabeled antibody or non-radiolabeled with high sensitivity. Cross-species reactivity of the antibody to antigen counterparts in mouse, rat, and monkey will be helpful for downstream validation experiments. Antibody binding selectivity over closest family members of target will be important. Of course, good affinity is defined as better than 1 nanomolar, and in vitro potency as defined as an IC50 range 10 nanomolars or better will be ideal. For immunization strategies, finding an experienced CRO service provider is crucial for the success of the project. Experience of the GenScript team suggests immunization with high-quality purified extracellular domain protein or a stable expression cell line to DNA immunization for single-pass membrane-bound protein usually assure good binders. For challenging targets like ion channel or GPCR receptors, DNA immunization, virus-like particle, membrane preps, or stably expressing cell lines will be used synergistically to boost the immune response. Intramuscular injection with or without adjuvant is usually sufficient for soluble protein antigens. The question of which antibody platform to move forward with is often a dilemma. The hybridomer-based approach is the most mature way to generate monoclonal antibodies. It is relatively inexpensive and easy to produce in small quantity. Human anti-mouse immune response is a substantial problem for therapeutic antibody purpose, so antibody immunization is a must-do before it can move further. Cavalate single-domain antibody is a unique antibody drug development platform. Conventional IgG antibody has 2 heavy chains and 2 light chains. It is composed of, in total, of 12 domains. Cavalate animals develop a unique class of heavy chain antibody during evolution. It only has 2 heavy chains and also missing the CH1. But this heavy chain antibody works very well, just like a conventional antibody, because it has an extended CDR3 loop, which partially surrogates the function of light chain. The first engineered single-domain antibodies are derived from the cavalate heavy chain. As its name implies, single-domain antibody, or SDAB, is comprised of only one domain, a 12 to 15 kilodalton molecule, much smaller than a conventional IgG. SBAB is a highly promising next-generation therapeutic antibody technology. The efficacy and safety have been validated in recent clinical studies. It is easy to modify for specific applications with short discovery cycle and low cost in production. The only drawback of this is that it has a short serum half-life, but GenScript has preparatory strategies to engineer it to have a much longer plasma half-life. Rapid immunization combined with phage display library is an efficient means to obtain high affinity antibody to various targets. Just like the mouse antibody, it needs full humanization. However, it is usually much more difficult. Compared to mouse antibody, to keep its original affinity and other biochemical and biophysical properties. We also provide a service, by the way. Human name antibody library is a shortcut to obtain human monoclonal antibody in a shorter amount of time. However, without in vitro or in vivo affinity maturation, its affinity is usually not as high, so thus limiting its application. It is more suitable as a way to develop reagent antibodies with limited budget. Drugs fail in the clinic for two main reasons. The first is that they do not work, and the second is that they are not safe. Lead optimization is a key to support therapeutic drug lead development. Humanization has become a standard and widely used technology to reduce immunogenicity of a therapeutic antibody initially derived from rodents. The process refers to the replacement of more than 90% of the rodent IgG sequence in the parental antibody molecule with human IgG sequence. In addition to humanization rodent antibodies, ongoing efforts in the field are also devoted to the conversion of other non-human antibodies into human therapeutics. And the humanization has been applied to therapeutic candidates derived from rabbit, chicken, and camel. Affinity maturation is often applied to antibody leads selected from a named human library using a display technology. These leads may have relatively low target binding affinity, but it can be enhanced using various affinity maturation technologies to reach a desired affinity range. The Fc region of an IgG1 molecule is a functional molecule, a molecular entity mediating first ADCC where binding to FCC receptors on natural killer cells. Second, CDC bears C1q binding. And the third, the increase in the in vivo half-life where binding to the innate receptor. Fc engineering is important to development of some drugs because it provides an antibody lead desirable features required for full functionality. Improved biochemical and biophysical characteristics such as expression, solubility, stability, aggregation prevention, glycosylation of the antibody is important for the development of downstream manufacturing, which is also important for development of commercial antibody drugs. Exploratory PK/PD and construction of bispecific or multivalent antibody also requires seasoned expertise. And GenScript provides all of the above-mentioned antibody optimization services based on a results-driven model. GenScript has licensed a proprietary screening platform called a placebo, which is short for fast screening for expression, physical properties, and affinity. This technology was licensed from the National Research Council of Canada. The platform provided crucial support to GenScript's antibody optimization services to become highly competitive. The platform has been used for most of our antibody optimization projects because it not only provides high throughput affinity ranking, hundreds of antibody variants using air coating 200, but also help to gate out the low expression clones, and the clones have poor biophysical properties such as aggregation. We also successfully use the platform to efficiently isolate SDAB as crystallization chaperones for ion channel packing. Candidate antibody validation is certainly an important step for antibody drug development. Before a robust antibody validation can be performed, it may be necessary to scale up antibody production by generating stable cell lines. In vivo efficacy assays like syngeneic tumor models may be needed to test the efficacy of immuno-oncology antibody leads. Subcutaneous xenografts or orthotopic models may also be of use and require validation as reference drugs. Candidate validation before a clinical trial is a complex, time and cost-consuming process. How we maximize the translation of efficacy into the clinic requires the consideration of various aspects and requires a seasoned R&D team with different technical backgrounds to work together. I won't get into too much detail on this today. Now I would like to introduce how GenScript's Discover biology team might be able to help you if you plan to develop antibody drug lead. As you may already know, GenScript is proud of providing one-stop antibody drug development services. Our gene synthesis, protein, and peptide synthesis departments provide us with a solid foundation for providing fast, economical, and high-quality services to our customers. Our in vitro and in vivo assay units provide sophisticated, customized assay systems to evaluate our antibody leads. Antibody and the protein engineering department, which I'm in charge, provides services like monoclonal antibody sequencing, epitope mapping, antibody characterization, antibody humanization, affinity maturation, SDAB development, rapid phage display, and all kinds of customized synthetic libraries. Hybridoma sequencing is the first step in antibody engineering and also protection against hybridoma loss, and it ensures that the hybridoma is actually monoclonal. Every year, we provide about 500 antibody sequencing services to big pharmas and biotech companies. GenScript antibody sequencing is competitive because we provide fast, inexpensive, and reliable services. Epitope mapping is a process identifying binding site of therapeutic antibody. It provides important information of antibody specificity and the selectivity features. Our proprietary flex peptide technology ensures quality of peptide library synthesis and fast assay development and fast delivery. In the past 5 years, we successfully delivered more than 50 antibody optimization projects, including humanization, affinity maturation, stability enhancement, and effector function enhancement. Other BU head, our BU head, Dr. Chen Cho, is the inventor of humanized anti-IL-4 monoclonal antibody and IL-10 receptor, co-inventor MCP1 Ig fusion protein solution. Our advisor, industry veteran, Dr. Leonard Presta, is the inventor of Herceptin and Avastin, key contributors to the success of many approved antibody drugs. Our antibody immunization services involve usage of comparative placebo technology for ensured protein stability and extensive bioinformatics analysis for selection of the best human recipient frameworks will be applied before library construction. We are the only CRO routinely provide guaranteed implementation or affinity maturation services. In other words, we provide a success-dependent optimization model. For most of the antibody optimization services, we will construct large variant library for the best antibody selection. These professional approaches ensure the quality of service and earn us a great reputation. To pursue success with our customer together, most of our services are highly customized, and our scientist team deeply involved into our customers' R&D activity. We provide our customer comprehensive roadmaps and options with detailed pros and cons indicated. As I introduced already, cavalate single-domain antibody represents a novel avenue for antibody drug development. It has many unique features and advantages over conventional antibodies. It has excellent affinity, water solubility, and a great tumor penetration ability. Recently, there are solid reports that SDAB can even pass through the blood-brain barrier, which opens the possibility of treating disease in receiving ends. SDAB has unique features such as resistance to the maturation by heat, detergents, and other nutrients. It has a wide range of applications in the field of biology, diagnostics, and immunotherapy. Although still novel, SDAB drug has passed the proof of concept stage. There are at least 2 SDAB drugs passed phase 2 clinical trial showing robust efficacy. Additional benefit of SDAB as future drug platform is that it can be manufactured in E. coli or yeast, which is much more cost-effective than mammalian cells. Currently, most of the pipeline SDAB drugs are produced in yeast. We have already over 5 years experience of SDAB discovery, and our expertise is comparable to the major leader SDAB drug developer. So as one of the leaders of this field, GenScript is no doubt the world's #1 CRO company providing SDAB discovery services. GenScript has also established its own extended technology to solve problems of short serum half-life, then impede their development. We have delivered many highly customized, humanized, or affinity-optimized SDAB to our customers from big pharma. For customers who want a very high affinity bandwidth, we provide SDAB that bind at a picomolar range. It's a high success rate. As one of the world's leading biology CRO, GenScript has established a comprehensive drug development platform to integrate antibody engineering and antibody production technology. Our scientists with extensive experience in antibody drug development provide secure services such as single-domain antibody generation, antibody humanization, affinity maturation, and antibody sequencing. To summarize, antibody-based therapeutics has entered the center stage of drug discovery. More and more biopharma companies devoted to biologics therapeutics, especially antibody drugs. Sophisticated and high standard discovery process of viable candidate from target validation to candidate identification are crucial to the success. GenScript, as one of the world's leading biology CRO, has established a comprehensive antibody drug discovery platform to provide reliable one-stop services. Our discovered biology team provides secure services such as single-domain antibody generation, antibody optimization, antibody sequencing, in vitro and in vivo pharmacology services, and many more. Thank you.
Thank you very much, Frank, for that wonderful talk. We would also like to thank everyone for taking the time to listen in on this webinar. We hope the information presented today has been useful to you, and you can apply some of what Dr. Fan has talked about today to your own antibody drug discovery projects. I would also like to let you know that there are 2 additional webinars coming up. If you are interested, please sign up at www.genscript.com/webinars. As mentioned at the beginning of the presentation, this webinar will be archived on our website, and links to resources will be posted after this presentation is over, so please visit our webinar webpage to take advantage of these resources. Thank you again.